right now, we possess maps of the mammalian brain that are, frankly, just unbelievable. I mean, they show the exact twists, the branches, the connections of individual microscopic cables.
Yeah, we have absolutely accumulated more raw neurological data than, any generation in human history.
We have, and yet neuroscientists are, well, they're virtually blind. Because it turns out having all these pristine map pieces is entirely useless if they're written in like 10 different coding languages.
Right, or formatted on completely different scales and locked away in isolated databases all over the internet.
Exactly. So our mission today for this deep dive is to look at how we finally solve that bottleneck.
It's the absolute definition of a data bottleneck, really. We've engineered the tech to digitize these brain-wide neuron morphologies, but the data is just siloed.
Right, on competing platforms, stuff like digital brain or mouse light.
Yeah, exactly. So a researcher looking for a specific projection pathway is essentially trying to navigate a sprawling invisible city, but every neighborhood uses a totally different map projection.
Which sounds like a nightmare. So today, we are digging into a groundbreaking Nature Methods paper. It details a platform called NeuroXiv.
Right, NeuroXiv.
And it's an AI-powered system designed to tame this wild west of brain data. And the goal for you, listening right now, is to really understand the mechanics of how AI is being deployed here.
Because it's not just storing this massive backlog of data, it's actively synthesizing it, translating it, and, actually understanding it.
Okay, let's unpack this. Where do we even start?
Well, before we can even bring AI into the picture to intelligently search the brain, the architecture requires a foundational step. You have to force all of this siloed data onto the exact same coordinate system.
Makes sense. I mean, you can't run a search algorithm through a library if every single book uses a totally different alphabet, right?
Exactly, or a different filing system. And the raw material we are dealing with here is neuron morphometry.
Which is just the physical shape of the cells.
Yeah, the highly complex 3D coordinates of the structural parts. So the soma, which is the cell body, the axon, which is the long transmission cable, and the dendrites, those receiving branches.
Okay, and the scale here is just wild. By aggregating from multiple independent sources, NeuroXiv has compiled, get this, 175,149 atlas-mapped neurons.
It's a staggering amount of data.
And within that data set, there are 19,406 fully traced axons. This is all derived from over 500 mouse brains covering 292 distinct cell types.
And processing just one 3D biological structure is computationally heavy. Processing 175,000 of them. That requires a brutal level of standardization.
So how do they actually do it?
It relies on two twin pillars.
Yeah.
The first pillar dictates how we describe the physical shape of the neuron itself, and that is the SWC format.
SWC, okay, break that down for us.
So SWC is a standardized 3D file format. Think of it as a way to convert biological spaghetti into a rigid mathematical matrix.
Biological spaghetti, I like that.
Right. Instead of dealing with images or loose 3D renderings, SWC represents a neuron as a series of connected points or nodes.
So if I'm visualizing a complex, branching dendrite, SWC is essentially plotting a dot at the start of the branch.
Exactly.
And it assigns it an X, Y, and Z coordinate, records its thickness, and then it crucially records a parent ID to link it to the next dot down the line.
Yes, that parent ID is everything. It turns a messy tree branch into a traceable numerical spreadsheet.
So it strips away all the visual noise.
It really does. It leaves a pure topological tree structure. So now the algorithm can trace the exact path of an electrical signal by just following those parent-child linkages in the data table.
Wow. Okay, but having a standardized shape is only half the battle, right? Because you still need to know exactly where that shape lives in the broader universe of the brain.
You do. Which brings us to the second pillar, and that's CCFV3.
Right, the Common Coordinate Framework version 3. This was developed by the Allen Institute, correct?
That's the one. The CCFV3 is a highly specific, standardized 3D grid system for the mouse brain. It divides the entire organ into these tiny 10-micron resolution blocks, or voxels.
10 microns, that is impossibly small.
It's at the cellular level, so every single neuron ingested into NeuroXiv, regardless of whether it came from a lab in Tokyo or, a server in Boston, is mathematically warped and registered into this exact same 3D spatial grid.
Okay, so if we force everything into that SWC format and then map it onto the CCFV3 atlas, we have essentially built a unified GPS system.
That's a perfect analogy, yeah.
So we went from people using latitude, longitude, and 20 paces past the old oak tree,
Right.
to everyone using the exact same standard coordinates on one universal globe.
Because without that universal globe, any AI analysis later on would just be comparing apples to abstract concepts. It wouldn't work. The rigorous standardization is what makes all the subsequent layers of the platform even possible.
Let me pause here and just lay out a quick recap for you listening so we have our bearings.
Good idea.
So the core problem is that neuroscience data is massively fragmented. Step one of the NeuroXiv solution is to ingest all that messy data and run it through a rigid standardization protocol. We use the SWC format to turn the 3D shapes into mathematical node trees, and then we use the CCFE3 brain map to anchor those trees to specific universal coordinates.
Spot on. And with the database unified, the challenge shifts from organization to navigation.
Right, because a single map containing 175,000 branching structures is practically opaque to the human eye.
It's just a dense forest. If a researcher wants to find a single biological pathway, writing the custom code required to filter that data is incredibly tedious. You have to be a coding genius.
And this is where the AI really steps in. The solution Neurofing introduces here is AIPM, or AI-powered open mining.
Yeah, AIPOM is brilliant. The concept is that a user can bypass complex query languages entirely. You just type natural English into a search bar.
So instead of writing code, a scientist can just type search for AIPOM neurons projecting to CP.
Exactly. They're asking the system to find cells originating in the primary motor area, that's the MOP, that send their transmission cables into the catoputamen, the CP.
And the interface just takes that plain language intent and maps it directly to the underlying metadata and those 3D coordinate filters we just talked about.
Right, and you can even ask broader questions like, search for neurons involved in behavior. And the system will cross-reference functional annotations to pull the relevant morphological data.
Here is where it gets really interesting, though, because, wait, I understand the appeal of natural language, but this brings up a massive technical hurdle.
You're thinking of the computational weight.
Yes, large language models are famously heavy. If I'm a researcher and I'm typing out natural language queries regarding my unreleased, highly sensitive data, sending all of that over the internet to a huge centralized cloud server introduces two major problems.
Latency and privacy.
Exactly. Cloud latency makes the process agonizingly slow. And second, it's a glaring privacy risk for proprietary research. Am I going to leak my lab's breakthrough to OpenAI?
It's a huge concern in the field.
Yeah.
But the engineers behind NeuroXiv bypass that bottleneck by completely rethinking where the processing actually happens.
Oh, really?
Yeah. Instead of relying on a massive server-side LLM to interpret every keystroke, they deployed a specialized natural language processing model, an NLP model, and a supervised decision tree that runs entirely client-side.
Client-side, meaning the intelligence required to parse the sentence lives directly within the user's front-end browser.
Exactly. It's utilizing your local machine's processing power. You don't have to send your search to a server to figure out what you're asking.
Okay, so how does that local NLP model actually break it down?
Well, it locally breaks down the syntax of your query. So it recognizes that MOMOP is a region, projecting to is a directional relationship, and CP is the target region.
And then what does the decision tree do?
The decision tree instantly translates that recognized intent into a lightweight database query string. The heavy lifting of rendering the actual 3D data happens via local WebGL technologies, so the query generation itself requires almost zero computational overhead.
That is incredibly elegant, and it explains the benchmark speeds they highlighted in the paper.
Oh, the speed improvement is massive.
Yeah, because the translation from plain text to database operation happens locally. It executes in just 2 to 3 seconds.
2 to 3 seconds.
Right. The researchers noted this client-side trick achieved a 12.3-fold improvement in speed over traditional server-side LLM methods.
Which is just a massive operational advantage when you're iterating through research questions.
Yeah.
But the privacy aspect is equally critical.
Right, because the data never leaves the laptop.
Exactly. The intent recognition and query generation happen on your machine. So the raw queries and any user-supplied proprietary data never go to a third-party AI company. It is completely secure.
Okay, so we've established how NeuroXiv solves the search problem. It gives researchers the intuitive interface of an AI chatbot with the speed and security of a local software program.
Yeah, it's the best of both worlds.
But searching is only one function. The paper details how AIPOM also generates these massive written data analysis reports based on these searches. And here's my major concern.
The hallucinations.
Yes. We all know large language models hallucinate. They routinely invent facts, they hallucinate connections, and they fail spectacularly at basic mathematics.
It's true. They're storytellers, not calculators.
Right. And if we're dealing with rigid, coordinate-based biological data, an AI hallucinating an axon connection that doesn't actually exist could derail a research project for months. How do they handle that?
Trusting generative AI with rigid numerical science is basically the Achilles heel of modern bioinformatics. But NeuroXiv attacks this by implementing a specialized architecture called the MOE framework.
Mixture of experts.
Yes, mixture of experts. It is a three-stage process designed to essentially put the generative AI in a straitjacket of empirical rigor.
A straitjacket for the AI. Okay, you just sold me on the elegance of a lightweight client-side search. Now you're telling me we are firing up a complex, multi-stage mixture of experts framework. That sounds like the opposite of lightweight.
Well, you have to separate the two functions. Doesn't running multiple AI models create a massive computational traffic jam?
Yeah, that's what I'm asking.
It would, if it were happening locally for every single search click. But the MOE framework is primarily utilized on the back-end pipeline to process, synthesize, and verify those deep analytical reports.
Yeah.
Or it's run asynchronously for really heavy synthesis tasks. So it separates the instantaneous local search from the intensive multi-model analytical synthesis.
Got it. Okay, let's break down how this straitjacket actually works. What is stage 1 of the MOE framework?
Stage 1 intentionally excludes generative AI completely.
Really? No AI at all?
None. A traditional rigid computer program takes the query data and generates a raw statistical report. It calculates the exact number of neurons, logs the specific brain regions, computes the precise average length of the dendrites.
So it produces a foundational document of pure mathematical fact with zero interpretation.
Exactly. The facts are locked in stone before any AI model is even allowed to be creative.
I love that. Okay, so once that baseline is established, what happens in stage 2?
Stage 2 is where NeuroXiv takes that raw data report and feeds it to 3 different independent large language models.
3 of them?
Yes, specifically, the framework utilizes QuenMax, Mistral Large, and GLM-4. These 3 models operate in parallel as independent data scientists.
So they aren't talking to each other?
No, completely separate. Their task is to analyze the raw statistics, identify morphological features, and draft narratives explaining the complex projection patterns.
So we basically have 3 different algorithms looking at the exact same numbers, trying to independently spot patterns and draft an analytical summary.
Right. But as you pointed out earlier, any single model might make a logical leap or hallucinate a trend or misinterpret a coordinate.
Which brings us to stage 3.
Yes, stage 3 introduces a final, entirely separate model. And for this, they use GPT-4o. This model acts as the ultimate judge.
It's essentially a courtroom trial.
That's a great way to put it.
Think about it. The raw data report from stage 1 is the physical evidence. The 3 smaller AI models from stage 2 are the expert witnesses presenting their competing theories to the court.
And GPT-4o sits on the bench as the judge.
Exactly, a ruthless senior editor.
So GPT-4o reviews the 3 varying reports. It rigorously cross-references every single claim against the rigid physical evidence from stage 1.
Checking their math.
Right. It identifies consensus among the models. It strikes any mathematical inaccuracies or hallucinations from the record. And then it synthesizes these surviving, verified insights into a single, concise, final report for the user.
And does it actually work? Like, did they test this?
They did. When the researchers benchmarked this MOE framework against us relying on a single generative model, the multi-expert approach consistently yielded higher accuracy and essentially eliminated the hallucination problem.
It's a brilliant failsafe. You're harnessing the pattern recognition capabilities of generative AI while completely neutralizing its tendency to invent data.
Exactly. You get the creativity without the liability.
Okay, time for another quick structural recap for you listening, just to make sure we've got all these concepts locked in.
Let's hear it.
For navigation, AIPOM gives us fast, secure, natural language searching by translating our queries locally on our own hardware. That's the 12.3-fold speed increase.
Yep.
And for synthesis, the MOE framework, the courtroom trial of AI models, ensures that any written analysis we receive is scientifically accurate and mathematically verified.
Perfect summary.
Yeah.
We have built the infrastructure. But the critical question now is how this unified, highly searchable data translates into actual biological discovery.
Yes, so what? of the entire project. What are researchers actually finding in these 175,000 neurons now that they can clearly see them?
Well, the paper highlights a really fascinating case study focusing on VPM neurons.
VPM, ventral postremedial nucleus.
That's the one. These are critical relay stations located deep in the thalamus. Their primary job is routing sensory information, particularly, tactile data from the face and whiskers in mice up into the cerebral cortex for processing.
Okay, so using the standardized NeuroXiv database, researchers were able to query hundreds of these VPM neurons across disparate datasets and trace exactly where they were sending their outgoing axons.
Right. And because the data was uniformly registered to that CCFV3 atlas we talked about, the system instantly highlighted a massive degree of nuance that we couldn't easily see before.
What kind of nuance?
The researchers identified 8 specific, distinct subtypes of projections originating from just these VPM neurons alone.
Wow. 8 subtypes just from one relay station. And the selectivity of how these subtypes wire themselves is fascinating. I was looking at this part of the paper.
Oh, layer skipping.
Yeah.
Yeah.
Because the cortex is traditionally divided into 6 distinct layers, right?
Layers 1 through 6.
And the researchers discovered that certain VPM subtypes project their axons up into the cortex, but selectively bypass specific layers. Like some subtypes project upward and entirely skip layer 5,
Yep.
and others project in and specifically skip layer 4.
And skipping layer 4 is a profound biological detail.
Explain why that is so profound.
Because in classical neuroscience, layer 4 of the cortex is widely considered the primary port of entry. It's basically the main reception desk for sensory information arriving from the thalamus.
So if an axon carrying sensory data is deliberately bypassing the main reception desk at layer 4 and delivering its signal directly to a different layer, what does that mean?
It implies a completely alternative processing hierarchy. It forces us to rethink how the brain prioritizes and routes specific types of sensory information. Maybe some urgent signals get to bypass the lobby and go straight to the executive floor, so to speak.
Visualizing that kind of layer-specific routing across hundreds of overlapping cells, that would take a human researcher months of manual rendering.
Easily. But NeuroXiv surfaces it almost instantly. And the platform also introduces another mechanism called spatial indexing, which pushes the discovery potential even further.
Spatial indexing. This is about finding physical neighbors in 3D space, right?
Exactly. Instead of just searching by cell type or brain region, a user can select a specific neuron and ask the system to index all the axon neighboring neurons or dendrite neighboring neurons.
So the database algorithm scans the 3D coordinates and returns any other cells whose branches physically intertwine or overlap with the target neuron.
Right, regardless of where their cell bodies actually originate.
It's like identifying who is physically shaking hands in 3-dimensional space.
That's exactly what it is. And if you find neurons that overlap physically, you can instantly query the database to see if they share similar overall shapes or if they target the same downstream regions.
So it allows researchers to investigate functional clusters and structural similarities based on pure spatial proximity.
And the velocity of these inquiries changes the fundamental nature of the research itself.
Oh, the speed benchmarks here are staggering. When the team compared NeuroXiv to an older browser-based platform called Digital Brain, they tested interactive tasks.
Like rendering specific analysis charts for CA1 neurons in the hippocampus.
Right. Yeah, exactly. And NeuroXiv consistently completed the task in 4 to 5 seconds.
4 to 5 seconds. And the architectural efficiency scales perfectly, regardless of how much data is loaded into the query.
Meanwhile, on the older platform, performing the exact same rendering task for the same CO1 neurons took over 80 seconds.
Over a minute and a half.
Right. NeuroXiv is operating nearly 20 times faster.
Which is huge.
If a researcher has to wait 80 seconds every time they ask a follow-up question, the creative train of thought just derails. You lose your momentum.
Absolutely. A 4 to 5 second response time transforms the workflow from a tedious data retrieval chore into an active, iterative conversation with the biology.
It really does.
And that speed is the ultimate payoff of the entire system. I mean, we spent the last 2 decades getting incredibly proficient at collecting data. But raw data sitting in a silo simply creates information overload.
Which is where we started this whole conversation.
Right. Knowledge only becomes valuable when it can be dynamically queried, rapidly visualized, and confidently synthesized. NeuroXiv is not just a digital archive, it's an active research partner that allows scientists to ask sharper, more complex questions.
Which is exactly why you, listening to this deep dive right now, should recognize the broader implications of a mouse brain database. Because it's not just about mice.
No, not at all.
The mechanisms we explored today represent a master blueprint for the future of knowledge management across almost every field.
Oh, absolutely.
Think about it. Whether you are analyzing chaotic financial markets or studying global supply chains or reviewing decades of legal precedent, the fundamental roadblock is identical. You have massive amounts of messy, unstructured data locked in isolated formats.
And the methodology of utilizing a localized natural language interface to unify that messy data and then deploying a strict mixture of experts' framework to synthesize it without hallucinations, that is a universally applicable architecture.
It really demonstrates how we will bridge the gap between human intuition and massive data sets in the coming decade. We are firmly transitioning from the era of data collection into the era of data synthesis.
We really are.
And as we wrap up, I want to leave you with one final lingering thought to mull over. We've seen how AI tools can seamlessly unify complex morphology and instantly flag hidden biological realities.
Like an axon stealthily bypassing layer 4.
Exactly. But right now, the AI is still just reporting the facts we ask it to find.
True.
What happens when we give these systems the autonomy to not just report the data, but to start independently proposing entirely new biological theories? Theories based on billions of microscopic, brain-wide interactions that human eyes and human minds simply lack the capacity to hold in their heads all at once.
That's the real frontier.
If the brain is the ultimate puzzle, we might have to accept that we aren't going to be the ones who finally put the pieces together.